welcome to today's webinar. This webinar is hosted by Jen and sponsored by Unchained Labs. Today's topic is Level Up Nanomedicine Development with High-Throughput Formulation Screening. Thank you so much for being here. I'm Uduak Thomas, Senior Editor at Jen, and I'm delighted to be your host for today's webinar. Let's meet our speakers. You'll be hearing from Dr. Anna Maria Macri-Pisticu, COO and co-founder of Nanoworx, Dr. Stijn Hofstraat, Senior Scientist at Nanoworx, Dr. Anne DeDrew, Senior Scientist at Nanoworx, and Dr. Ben Nappet, Senior Product Manager, Sunny Suite at Unchained Labs. We'll get to their presentation shortly, but first, a bit of background on today's topic. With countless nanoparticle formulation options to explore, researchers need a way to quickly identify scalable nanomedicine formulations that deliver their payload efficiently and consistently. Traditional approaches are slow, resource-heavy, and often make it hard to see which candidates truly stand out. Using scalable high-throughput tools from Unchained Labs such as Sunny Suite, Nanoworx works with scientists to explore the broad design space of nanoparticle optimization, ensuring high precision and minimal human error throughout the formulation process. So in this webinar, our expert speakers will show how Nanoworx combines technical expertise with scalable workflows to support the seamless development of nanomedicines from early discovery and design to large-scale production. During the webinar, you will learn how the company performs nanoparticle library production and screening with Sunscreen, fine-tuning and scaling with Sunshine, sample workup with Big Tuna, and batch production with Sunbather. Key takeaways include the integration of scalable manufacturing, precise liquid handling, and advanced analytics in a single pipeline, lending expertise with infrastructure to support seamless nanomedicine development from concept to lead material production, and minimizing development risks, reducing time to market while ensuring consistent quality. Today's presentation is going to be followed by a live Q&A. At any point during the webinar, if you have a question, please enter it in the Ask a Question panel on your screen. All right, let's get started.
Hello everyone, thanks for taking the time to join us today for this webinar, where we'll be talking about how you can level up your nanomedicine development with high-throughput screening. When it comes to establishing a capability for the development of nanotherapeutics and nanomedicines, it can be hard to know where to start, but you do generally have a good idea of what it is that you need. Well, firstly, we need a capability to make, purify, and characterize our particles on a daily basis. During the early stages of formulation discovery, this is going to involve trying out lots of different payload and formulation combinations. And so an ideal process is going to minimize the cost per experiment by limiting the reagent consumption per experiment, as well as limiting consumables. In addition to this, the automation of the process is going to save a ton of time and improve the consistency of our results. And a final consideration it's important not to overlook is that eventually a successful candidate nanoparticle formulation will need to translate to a continuous manufacturing process. And so it's key that particles that are made at small volumes can also be produced in the same way for larger volumes later down the line. To make a lipid nanoparticle, to use a lipid nanoparticle as an example, obviously we could also make liposomes, polymer nanoparticles, or other types of encapsulation materials. We need to take our basic LNP ingredients, a mixture of lipid types and a candidate nucleic acid payload, and convert them into a highly complex particle structure in a way that ensures that every particle is similar in that structure and in particle size. Fortunately, solvent injection provides an easy way to achieve this, with the lipid molecules themselves doing most of the hard work via a self-assembly process. By mixing up our lipids at a desired concentration and ratio into a solvent such as ethanol and our nucleic acid into an acidified aqueous buffer, nanoparticles will self-assemble when those two solutions are mixed. And that's generally done at a 3:1 ratio in a flow-based process. The way these two fluids mix together is critical to forming a high-quality nanoparticle formulation. The particle self-assembly is driven by the changing polarity and environment surrounding the molecules, and is influenced by things like the mixing geometry, the flow rates and ratios used, as well as the formulation itself and the pH at which all of this takes place. Mixing at larger scales results in regions of high concentration gradients and poor mass transfer, and ultimately a slow and uncontrolled mixing process. Microfluidics creates an environment in which mixing takes place reproducibly in a matter of milliseconds, mixing together very small volumes at any given moment, offering an ideal way to produce nanoparticles with tightly controlled properties. Most of the nanoparticle synthesis systems available for synthesis and characterization use a one-at-a-time approach, with each formulation prepped and measured individually. To screen large numbers of formulations, automation using a well-plate format combined with a low reagent consumption for both synthesis and characterization unlocks the ability to process significantly more formulations in a day than is possible with one-by-one microfluidic approach. Sunscreen and Stunner offer exactly that: high throughput, low volume synthesis and characterization of nanoparticles on a 96 well-plate format. Sunscreen processes a unique formulation every 3 to 4 minutes by combining the lipid in ethanol from one well and the RNA in its aqueous buffer from another well plate, with input reagent volumes ranging from as little as 100 microliters up to 2 milliliters. A typical low-volume formulation at a 3-to-1 ratio uses 400 microliters of reagent. With a CV in particle size of less than 1% across 90 repeats, even with some bad formulations mixed in, as shown in the graph at the top, you can screen formulations with confidence and create large data sets for detailed insights on how your formulation changes will affect the particle properties in the final particle. The data shown at the bottom shows 32 triplicate experiments that were designed to explore various different lipid combinations and were all run and characterized in a single day, allowing for rapid iteration of formulations to then focus in on a winner. And all of this is done using our glass microfluidic sunnies, which come in a range of mixing geometries to offer maximum flexibility for the mixing process. The sunny itself is cleaned in place after every experiment by the driving fluids, which are used to push the reagents through the system, and that's typically ethanol and the aqueous buffer, but with no expensive materials in them. And the sunny is fully reusable to eliminate per experiment consumable costs, making sunscreen screening of either hundreds or thousands of formulations a much more realistic prospect. Now, once you've made all of these particles, there are a lot of questions that need answering in order to help identify promising candidates for further downstream testing. Answering all of these questions can often involve multiple different instruments and several fiddly assays. A Stunner takes the hassle out of particle characterization by providing RNA and particle quantification, encapsulation efficiency measurements, and sizing data and information on the presence of aggregates, all in the same readout. Here you can see what loading a sample looks like for Stunner. In 2 wells, we can already see that we've added just 2 microliters of, in this case, fluorescein dye. To load another sample, you simply pipette into the next well 2 microliters of sample, and that wicks into the plate. And that's all the reagent needed in order to characterize your sample. What Stunner then does is uses 3 different measurement techniques to characterize the 2 microliter sample. It uses UV-Vis, rotating angle DLS, which looks at both forward and backward scattering angles, and also fluorescence. This then gives you sizing and particle concentration from the DLS, plus information on aggregates from those forward scattering angles, total RNA measurements from UV-Vis, and the free RNA quantification from the fluorescence data. And those last 2 points together mean Stunner can calculate an encapsulation efficiency value to display in the report. For real samples, the results compare closely for encapsulation efficiency with the standard ribogreen assay, but with significantly less reagent and time required, and no need to do the extra step of breaking open the LMPs with a surfactant. The final need was the ability to scale. Now, SunnySuite instruments do this by using exactly the same pumping technology on each instrument, as well as the same microfluidic mixing geometries. This means that when handing over from screening to process development, or say from preclinical to clinical studies, the production process of the particles is identical, and no optimization is therefore required when moving between instruments. Sunshine offers both the low-volume automated mode, like Sunscreen, and continuous production, like our GMP Sunbather, and is able therefore to bridge the gap between those two instruments. The automated low-volume capabilities of Sunshine allow rapid identification of optimal process conditions for your chosen nanoparticle formulation, like the data shown here in the top right, testing 4 different flow rates per run with consistent triplicates. And the continuous production capability, shown in the bottom right, allows you to produce larger volumes of material for preclinical testing and to test manufacturing runs ahead of GMP implementation with Sunbather. Nanoworx have implemented each of these instruments into their laboratories, as well as Big Tuna for high-throughput buffer exchange of nanoparticle formulations. I'm excited to now hand over to Anna to show us how these tools are being put to use in combination with their leading expertise to facilitate the design, development, and production of advanced nanomedicines.
Thanks, Ben, and hi everyone, and a very warm welcome to today's webinar from all of us at Nanoworx. We're genuinely excited to be here, sharing some of the work we are doing, and most importantly, showing how we can partner with you to accelerate the development of your nanomedicines. Nanoworx is a specialized contract research organization fully dedicated to nanomedicine. We work with clients to design, optimize, and test advanced nanoparticle-based therapeutics and delivery systems. Although we are a young company, just about a year into full operations, our services are already comprehensive and fully up and running. I'll walk you through them in just a moment, along with real examples of how we're helping clients move their projects forward more efficiently. Our team is right here on the slide. I am Anna Maria, Chief Operating Officer at Nanoworx. Joining me here today are Anne and Stein, both key members of our scientific core. All 3 of us, together with Ben, will be available right after the webinar for a dedicated Q&A session. What makes our team effective is our complementary mix of expertise and hands-on nanomedicine experience. Our scientists are trained at the PhD level in areas like RNA delivery and advanced formulation, and we combine this scientific depth with strong project management, commercial awareness, and clear communication. This allows us to work closely with clients and consistently deliver high-impact results. Let's take a quick step back for a very brief introduction to nanomedicine. Nanomedicine is about engineering materials at a nanoscale to improve how therapies interact with the body. At the heart of this field are nanoparticles, which act as tiny engineered carriers that deliver therapeutics to their site of action while protecting them. By doing so now, nanomedicine enables more effective treatments with fewer side effects, ultimately improving patient outcomes and expanding what's possible for complex diseases. Moving on, let's talk about the real potential of nanomedicine and why that potential still isn't fully realized. This field has more than 5 decades of scientific innovation behind it. Labs around the world have developed countless proofs of concept. The therapeutic promise is truly enormous. Yet, despite this momentum, many of these innovations never make it past the proof of concept stage. The reason is not lack of scientific creativity. The real barrier is a know-how and manufacturability challenge, the practical challenge of turning a clever idea into something reproducible, scalable, and ultimately suitable for clinical development. Bridging that gap requires specialized expertise, dedicated infrastructure, and workflows that are built for systematic optimization. That is exactly why Nanoworx was created, and that is to accelerate the journey from a promising concept to an optimized, translationally ready nanomedicine. It's a combination of expertise and the state-of-the-art infrastructure needed to execute it reliably. In the next slide, I would like to show you exactly how Nanoworx does that. Let's start by looking at how nanomedicine development typically happens today. A concept emerges, for example, from academia or a startup that non-optimized now concept is then moved directly into a CMC trajectory. But here's the problem. The difference between a proof of concept nanoparticle and an optimized nanoparticle can be dramatic, and that is sometimes orders of magnitude in efficacy or biological performance. When you carry an unoptimized system into CMC, it is likely to fail somewhere along the developmental pathway. It's not the science that fails, it is the lack of optimization before translation. Now, here is where Nanoworx fits in. Instead of pushing a proof of concept directly into CMC, we take that early concept and run it through a systematic, high-throughput library screening approach, supported by our automated microfluidic formulation platforms and advanced analytics. This step is now crucial. It allows us to explore the design space efficiently, identify optimal compositions and process parameters, and arrive at a version of the nanomedicine that is robust, reproducible, and ready for scale-up. Only after that optimization do we hand the formulation off to CMC development. This dramatically reduces risk downstream and gives our partners a formulation with a much higher probability of success. At Nanoworx, we create value for our clients in two complementary ways: through our deep domain knowledge and through advanced infrastructure. You can kind of see it as two modules. The first module is knowledge-driven. We are located on the campus of the Eindhoven University of Technology. We leverage the wide range of expertise within Nanoworx and that of the university ecosystem in which we are located. This now allows us to help clients design and prototype nanomedicine concepts from the ground up. This is particularly valuable for clients who may be new to nanomedicine, as we can guide them through formulation choices, platform selection, and early feasibility, turning initial ideas into workable, well-designed prototypes. The second module is infrastructure-driven. This is where Unchained's Sunny suite comes in. We use microfluidic and analytical equipment to efficiently build formulation libraries and systematically optimize these products. This enables rapid iteration, robust datasets, and continuous improvement, all within workflows that are designed for scalability and translation. However, delivering this level of service extends beyond microfluidics alone. Unchained Labs' advanced platforms are central to our formulation capabilities, providing a robust core that is further enhanced by complementary technologies. To support our clients, we also integrate advanced analytics, allowing us to characterize nanoparticles and extract meaningful data. In addition, we use automated liquid handling to enable high-throughput workflows. Together, this broader infrastructure, combining formulation, analytics, and liquid handling, allows us to go beyond simply running experiments. It enables end-to-end workflows that are robust, scalable, and tailored to real client needs. This slide here highlights our core service offering, but it's important to know that we're not limited to these building blocks. We work closely with each individual client to tailor the scope to their specific goals and constraints. We typically support projects from early concept and prototyping through fast, data-driven library screening into lead scale-up and advanced characterization. By combining flexible services with close collaboration, we help our clients move faster, make better informed decisions, and reduce risks across their developmental trajectory. At Nanoworx, we support essentially any self-assembled nanomedicine platform that can be produced using microfluidics. That is from simple to input systems to more complex multi-input configurations. You can see a few examples of these platforms illustrated on the slide. Today, I'll walk you through 3 short case studies to show how this support works in practice. I will start with the first case study, which focuses on lipid nanoparticles, or LNPs. As Ben previously explained, these are lipid-based nanoparticles designed to encapsulate and deliver a nucleic acid payload. To show how we support LNP development in practice, I'll first walk you through our standard robust LNP production workflow. Our workflow begins with preparing the input components required for the LNP formulations. All inputs are prepared in a fully automated manner using liquid handling, which ensures high accuracy, excellent precision, and strong reproducibility from the outset. Once the inputs are ready, the LNPs are formulated using the Sunscreen platform. Following formulation, we perform DLS analysis on the scanner to assess key quality attributes. You can see on the slide that all 20 individual formulations show the same diameter and PDI. This initial formulation step already delivers highly reproducible results with a relative standard deviation below 2.5%. However, at this stage, the particles are still considered crude and have not yet been purified and still contain organic solvents from the formulation process. So to reach a final application-ready formulation, buffer exchange is required, and that is why we've integrated Big Tuna directly into this workflow. Here on the left, you see the same data as before, but aggregated into two bars for easier comparison. This is our complete workflow for generating LNP libraries, now incorporating Big Tuna for automated buffer exchange. After buffer exchange, the final LNP product is analyzed again using the scanner, and depending on the needs of the project, we can also offer more in-depth analytics. We have optimized our workflow in such a way that the buffer exchange step does not negatively impact particle size or polydispersity index, as can be seen from these graphs. The RNA recovery also remains above 95%. Altogether, this demonstrates our ability to robustly and efficiently generate high-quality LNPs. The Sunny Suite is designed with translatability in mind. In practice, this means that scaling is straightforward. In-house, we've validated seamless translation across platforms, from the Sunscreen to the Sunshine and ultimately to the Sunbather. This spans production volumes from microliters to milliliters and all the way up to liters. For these LNPs, both particle size and PDI remained consistent across all three systems, clearly demonstrating that our workflow is not only robust, but also fully scalable. Next, we move into our liposome showcase. Liposomes are spherical vesicles composed of one or more phospholipid bilayers. They can encapsulate drugs or biomolecules either within their aqueous core or embedded in the lipid membrane itself. Unlike now LNPs, liposomes are typically bilayered structures. They do not contain ionizable or cationic lipids designed for nucleic acid delivery. Here, we demonstrate our ability to reliably produce these classic liposome formulations using our workflow. The data shows excellent reproducibility in particle size, along with high monodispersity across runs. To further confirm that we are producing true liposomes, we performed cryo-TEM imaging of these formulations. These images clearly reveal the characteristic bilayer morphology, as you see on the slide. Now, having established and validated this liposome production workflow, we then applied it all together with one of our partners to produce a library of liposome-like nanoparticles. In this specific case, a subset of 16 distinct compositions of liposome-like nanoparticles was selected for formulation, represented here by each individual color. Each composition was produced with 6 replicates, resulting in a robust and statistically meaningful dataset. What's particularly illustrative here is the diversity that emerges even within this relatively small design space. Even within this limited set of 16 formulations, these cryo-TEM images reveal a wide range of morphologies, bilayer organizations, and overall particle architectures. This structural diversity aligns well with the variation we observe in the measured physicochemical properties. With only 16 compositions, we already see a broad range of outcomes across key characteristics, highlighting how powerful systematic formulation screening can be. To close, I would like to move away from the well-known nanoparticle platforms by showing you an end-to-end client project built around library screening of a novel proprietary nanomedicine platform. We started with technology transfer, where we selected the right chip and flow conditions and demonstrated feasibility early on, helping to de-risk the project from the start. Next, we moved into process optimization, setting up a smooth and reliable workflow that made efficient library production possible. Once the workflow was established, we focused on process validation, defining critical parameters and quality attributes to ensure the process was robust and consistent. With all that in place, we ran the library production, and all of this came together in the results and reporting step. This data that I show here is real but anonymized and randomized, and it illustrates the depth of insight generated from the study. Ultimately, this translated into very clear value for our client. We dramatically shortened development timelines and moved from early exploration to actionable conclusions much faster than with traditional approaches, while at the same time screening large formulation libraries enabled a much deeper and more systematic optimization of the platform, revealing trends and design choices that would otherwise have been very difficult to uncover. Finally, now, by establishing a well-defined and validated process early on, the project was de-risked from a translational perspective, providing the client with a clear and credible pathway towards scale-up and eventual GMP production. In summary, nanomedicine needs more than great ideas and needs systematic optimization prior to translation. At Nanoworx, we combine deep expertise with state-of-the-art equipment infrastructure to do exactly that, and that is to enable faster development, stronger data, and lower risk. Most importantly, we do this as a flexible, hands-on partner, tailoring our approach to each client's goals. With that, I'd like to thank you for joining us here today. If anything you've seen resonates with you, your work, or raises any questions, we'd be very happy to connect and explore how we could support your program. And we're, of course, happy to answer any questions during the Q&A session. Thanks.
Many thanks to our speakers for their informative and interesting presentations. We're going to begin the Q&A session in just a few moments, so please submit your questions now if you haven't done so already. Before we start the Q&A, a reminder that this webinar will be available to view on demand in the next 24 to 48 hours, so please share the link with your colleagues who might not have been able to tune in today. And a very warm welcome to each and every one of you. Thank you so much for being here. We hope you enjoyed those presentations. And we hope you've got some great questions for our speakers. So as a reminder, if you have a question, please feel free to enter it in the Q&A panel on your screen. We've got lots of time for questions, so please feel free to get those questions in. I'm going to go ahead and get started with the questions that I see here on my screen. And Anna Marie, I'm going to come to you first. How do you handle the IP of clients?
Yeah, that's a great question and a very interesting one. Basically, protecting our clients' intellectual property is really, really important and fundamental to how Nanoworx operates. That being said, all the IP that clients bring into the project, and I think that's important to mention, remains fully theirs. But on top of that, anything we develop that is directly related to this background IP is also assigned entirely to that client. So from the moment a project begins, we treat all materials, data gathered, and know-how as confidential and protected, and we have processes in place for that. So our role is to generate high-quality science, not to claim any ownership. And we hope that this gives our clients the confidence that they can share early-stage ideas or proprietary technologies with us without any risk.
Excellent. Thank you so much. I'm actually going to stay with you for this next question, Anna Maria. Can you help interpret data or make strategic decisions? Can you help with those things?
Yeah, we can basically assist as much as the client desires. This is like the summary of it. This really spans from formulating the research objectives, so really early on, to the design of the experiment in the experimental design space, to data interpretation, project management, but also any follow-up actions.
Excellent. Thank you so much. Next question I have, and Ben, I think this one's for you. What type of particle can you make with the sunscreen?
Yeah, so we have a lot of data, and there's been a lot of content in this presentation about LNPs, and so of course all sorts of different LNP formulations are frequently used with the instrument. But we also have customers and users working with liposomes, polymer particles, hybrid structured nanoparticles that use a mixture of both, and that can sometimes involve some solvents and things which are a little bit more aggressive, but the system's suitable to handle that, and that allows you to work with polymers and lipids in a combinatorial way, which is unique. And in addition to that, there's interesting publications out there using things like chitosan and other types of more novel materials. So really, if you can flow those two fluids together and produce that particle in a precipitation process, then the equipment can be used for that.
Excellent, excellent. Thank you so much. And to the audience, once again, if you have questions, please feel free to send them through. We've got time for lots of questions. Anne, I think this question's yours. What is the scale of the libraries that you can make?
Thank you. Basically, we can create libraries at any scale, more or less. So it depends a lot on the design of the library. It's mostly a design choice. So maybe as an example, we've done libraries up to about 100 nanoparticles. It's one of the projects we're working on now. So it's basically as much as you need to get to the answer to your research question. So it's mostly a design choice. Yeah.
Excellent, excellent. Thank you so much. Ben, back to you for this question. What chip do I use for typical LNPs?
Yeah, we offer a range of different mixing geometries. So there are 9 in total, and usually my go-to mixer chip that we use for a lot of our particle formulation, for a lot of LNP work, is something like our 190 micron junction cross 190X chip design. So that enables us to have really controlled mixing of fluids, and we can use a range of different flow rates for that, so you've got a lot of control. But then, as you start to explore different formulations, and especially as you start moving towards that process optimization step with Sunshine, you can start exploring some of those other geometries and take advantage of some of the more nuanced geometries we offer too.
Excellent, excellent. And next question I have, I think, is also again for you, Ben. Do experiments have to be planned in the software for Sunscreen?
So the software for Sunscreen and Sunshine, both of those come with an embedded and validated spreadsheet that you can simply export from the software, and then you can plan all of your experiments inside that spreadsheet. And every row that you complete and enter in the values and parameters for your experiment, there's a validation check and calculations to make sure that your experiment is within the parameters the instrument can run. You can copy those all in, and then there's also things like a little summary calculation telling you how much time it's going to take to run those experiments, things like that. You just import those back into the software and click go, and you're ready to get started.
Excellent, excellent. Next question I have, I think this one's for Stein. How do you quantify RNA?
Well, thanks for the question. So the data on the slides that we've shown in the webinar shows data obtained with the stunner. So that shows total RNA. So not the stunner AF, I'll mention, but we've also in-house validated the ribogreen assay and automated that. So typically we use a combination of those two.
Excellent, excellent. Thank you so much. And Anne, this next question I think is also for you. What additional analysis can you do?
Yeah, so we can basically do a large range of analytical techniques. So we in-house have a couple of different things available. Some of them were mentioned on the slides. We also have some partners that we can, partner with to develop some other methods. So basically, it also depends a little bit on what the client would need. So anything is up for discussion, basically. So we can co-develop new methods. We can provide methods that we have developed in-house. So it can be anywhere from physicochemical analysis with DLS or molecular quantification on HPLC or even cell assays. It's very diverse.
Excellent, excellent. And to our audience, thank you once again for sending in questions. Please keep them coming. We've got lots of time. Stein, I think these next two ones are going to be for you. How do you do buffer exchange?
So for the smaller volumes, we typically use the Big Tuna, especially when we use a library production workflow because it has a nice plate format. But when we scale up and we go more to the Sunshine or Sunbather machines, we go to TFF. We have used spin filtration and dialysis, but that's more in a tech transfer setting. But typically, we like to go to the systems mentioned before.
Excellent, excellent. And the next question I have, what types of nanomedicine can you work with?
That is a very broad question. So typically everything that can self-assemble and be made with microfluidics can be produced. So that can vary from lipids to polymers and let's say everything in between. So we have validated the most common ones, but we are always open to take on challenging projects.
Excellent, excellent. Next question I have is cross-contamination between collected samples and issue. I think this one might be yours, Ben.
Yeah, so the instrument is cleaning. So for Sunscreen, the instrument's cleaning between every individual experiment, and the same for Sunshine. As an automated process that uses the driving fluids that are moving the fluids around the system to clean out the flow path. And that includes cleaning sample loops, cleaning the needle for aspiration, both on the inside and the outside. And so those have been tested and validated to remove your typical contamination. But you also have the ability as a user, if you want, in that spreadsheet I mentioned earlier, you can toggle between a low, standard, and high wash rates. If you want to be more aggressive with the washing, you can instruct the system to spend a little longer washing in between each experiment. And we have plenty of data that we can share if you're interested to kind of validate that cleaning process as well. Yeah.
Excellent, excellent. Thank you. Next question. I think this one's going to Anne. What volumes can you produce?
So like we showed in the webinar, we can produce with the Sunscreen, the Sunshine, and the Sunbather. So that means that we can scale from microliters, let's say a couple hundred microliters, all the way to big batches of liters and anything in between.
Excellent. Excellent. And Anne, I'm actually going to stay with you for this one. Can you handle customized or unconventional projects?
Yeah, sure. So we've worked with clients that have some proprietary nanoparticles, for example, that are a little bit different from the most standard LNPs. And we basically work together with these clients with with our flexible workflow, with knowledge that we have here within the ecosystem to try to get the best results from them as well. So we can definitely work with more unconventional nanoparticles or other projects. Yeah.
Excellent, excellent. Thank you so much. And thank you once again to the audience for the questions you've sent in. Please do keep them coming. This question, I think it's got a two-parter. How do you convert the formulation ratios you want to test into pipetting volumes? Do you use a specific software or tool for that? And then also, at what stage, if any, do you use machine learning tools in the workflow?
So I can answer the question. So we have developed in-house methods to make efficient conversions from ratios to volumes. But we can also input our clients' desires at multiple levels. So some prefer to give volumes, some prefer to give ratios. So we have our in-house methods for that, and we can always discuss with this particular person asking the question if they want to find out more.
Excellent, excellent. Thank you so much. Next question I have, I would like to know what are the limits of what the limit of detection of the stunner detector is for the different characterization detectors it is based on. Hopefully, I read that correctly. And then in terms of mouths, which is the sensitivity in terms of size for QC? And then a third question, can you differentiate between a few nanometers of difference with accuracy? So we've got sort of three
I can
questions in one.
tackle those. So, so with Stunner, so the limit of detection for things like RNA that you're trying to measure, typically we're quantifying down to around 5 micrograms per milliliter. And then in terms of particle concentrations, we can deal with a really wide dynamic range of particle concentrations. But one of the nice things about that, the resolution question, is because Stunner is using multiple angles, so we rotate the detector within Stunner to measure different scattering angles of light from the sample. It gives us an ability to probe the different biases between, say, smaller particles, which tend to scatter differently to larger particles. And we can then take all of that data and come together to create an angle-independent measurement of the particle size. As well as give you really detailed readouts on things like the presence of aggregates. So it's extra sensitive to that and can help you to really identify the most robust samples that you have. So yeah, we're able to tell the difference between fairly close populations of particles and yeah, distinguish those. We're also, in terms of the dynamic range of detection quantities, we can go to high concentrations as well. And so one of the challenges sometimes when you're trying to use something like UV to measure concentration of something like RNA in a nanoparticle sample is the turbidity. And so we get around that by having these really tiny path lengths through our turbid samples of LNPs that allows us to extract quantitative RNA concentrations even when your particle concentration is high. So yeah, that's a nice benefit too.
Excellent, excellent. Thank you so much. Next question I have, is sunshine useful for a two-step microfluidic process? So for example, an in situ conjugation of a biomolecule post-LNP synthesis.
Yeah, so you have a couple of options there. So the system comes with inline dilution. So we have one of the nice flexibilities around the sunnies is you can have either a 1 sunny setup or a 2 sunny setup. So you could have, say, one, commonly for LNPs, you might use that first junction to make your particles. In the second junction, you introduce a diluent. But it's entirely possible to use a third reagent solution to pump into that diluent channel if you wanted to do multi-step processes. The other thing is it's super straightforward to reload the sample back in the system and run it as a second pass if you wanted to tackle it that way as well. But yeah, I'd love to jump on a call with whoever asked that question and go into more detail on that.
Absolutely, absolutely. Ben, I'm actually going to stay with you for this next question. Does Stunner AF measure EE% on each sample?
So the way that Stunner is going to measure your encapsulation efficiency is by having a few wells dedicated to a concentration calibration curve, and then your sample wells are going to be in pairs at least, and you can always duplicate and triplicate those. One where you have your sample as is ready to measure, and where we're going to measure the total RNA concentration with UV-Vis, and then another well in which you've added a small amount of the ribogreen reagent to allow us to use the fluorescence detector to pick up the free RNA. And it's the combination of those two data points that gives you your answer. But it does mean that you can stack very large numbers of samples on a single plate, and so you can make this a lot higher throughput than a standard ribogreen assay. It's a much reduced assay. And yeah, the hands-on time is significantly less with a lot less pipetting, because you don't have to pop open all those particles to release the cargo to calculate your free RNA.
Excellent, excellent. Next question I have, what is the biggest scale that can be manufactured if you detail the liters term, and what is the highest total flow rate that can be used? So a two-parter there.
So I'm guessing that's focusing on, so Sunbather has each of the microfluidic channels on Sunbather is a replica of the channels on our Sunscreen and Sunshine systems. So you get that consistency of throughput from one system to the next. And so each of those channels is up to 10 milliliters a minute. So depending on your flow conditions and flow rates, you can use those channels together to generate your max total flow rate. And then as a way of scaling that initially to get to larger volumes, obviously you can run that system continuously to produce tens of liters of formulation. If you need to shorten that time frame down, you can run multiple Sunbathers in parallel, and they're designed to be used in that way, and we have customers doing that already.
Excellent, excellent. Thank you all so much for once again for great presentations and for taking time to answer quite a few questions today. We really do appreciate you. And to the audience, thank you so much for being here. Before we go, I did want to let you know this webinar is going to be available to view on demand. That should be in the next 24 to 48 hours. So what's going to happen is whatever email you use to register for this particular event, you're going to get a link at that email and you'll be able to click on it and come back and watch the entire webinar through from beginning to end. And we invite you also to share the link with your colleagues. Oh, I see we might have a couple more questions that have popped in at the last minute. Maybe we can take at least these two that have come in before we do have to bring the webinar to a close. Question, can stunner be useful to quantify the degree of conjugation of a biomolecule onto LNPs?
Yeah, so that's a great question. We do have people using that. So you can add things like custom analytes into the stunner software. So if you have a specific biomolecule that you're looking, or a marker that you're looking to detect alongside your LNP and alongside your RNA, we have tools to help you do that. And yeah, we'd love to talk to you about that more.
Excellent. And this next question kind of gets at that last point, Ben. How can folks reach you if they have more questions for everyone?
Yeah, absolutely. LinkedIn, I think we're all around on LinkedIn. And certainly if you tick that box, we'll be able to get in touch with you and follow up with your questions after the webinar as well. So look forward to speaking to you more.
Yeah. And
On
Anna Maria?
behalf of Nanoworx, the same. You can contact us either via LinkedIn, email, or at the contact@nanoworx.nl. We're happy to talk to you.
Excellent. Excellent. Wonderful. Well, once again, thank you all so much. And again, I invite our audience, when you do get that link, please do share it with your colleagues who might not have been able to join us live, but who you think would benefit from everything and who might have questions of their own that they would like to get in touch and ask. So once again, many thanks to each and every one of you, our speakers, for being here and for taking time to answer questions. I'd also like to thank today's sponsor, Unchained Labs. Thank you so much. And I'd like to shout out the superb multimedia group at Sage for all of their support. Thank you so much, guys, for making my job really easy. And most of all, thanks to every single one of you for watching today. For everyone at Gen, I'm Udwak Thomas. Thank you and goodbye for now.